Well, hi to everyone. Today we have with us a very special guest, and I want to give a very short introduction. 2 or 3, maybe 4 months ago, when we started to organize this series of lecture, I called Eduardo Satara, that I think that most of you know him, and I said, Edu, could you invite a referent in Evo Devo for our classes? Because Edu Edu did a very beautiful doctoral thesis on the subject, and he's a regular guest in our classes, in our evolution classes all the years. And he worked with amazing people in the USA. And 2 days later, he called me and said, Armin Moczek could give us a lecture. What do you think? And that was an answer that doesn't have any sense because it was Armin Moczek. And well, today we have Armin here with us, which is an honor for me. But I think that Edu could give us a much better presentation that I could do about who is Armin. So Edu, it's all yours.
Well, hello, welcome everyone. Bienvenidos a todos. Vamos a hacer una bienvenida muy breve en castellano y después voy a pasar al inglés. De hoy es nuestra primera charla del ciclo en inglés. Acuérdense que esto va a quedar grabado, así que no entren en pánico si se pierden algo, que esto después va a mantenerse en YouTube. Vamos a ver si le ponemos subtítulos y demás. I want to introduce you today to Dr. Armin Mochik. He is a professor in Indiana University at Bloomington. He's a very well person in the field of Evo Devo. He was also one of my postdoctoral advisors. I worked in his lab for 2 years, and 2014 and 2015, and we keep collaborating. Let me tell you, Armin is German. He was born in Germany. I think he hails from the area of Bavaria. He got his master's degree at Julius Maximilians Universität in Pittsburgh. He worked then at rainforest ecology. He was looking at what patterns the diversity of arthropods on tropical forests. And then as a part of his master's and some exchange, he went to Duke, and there he met Fred Nyhout, who was working, among other things, on the Evo Devo butterfly wing pattern, and he really liked that kind of work. So he ended up going to the US, to Duke University, to make a PhD with Fred Nyhout. He did not work on butterflies. He ended up doing something else. But he really liked the field, and he remained working on Evo Devo topics. And he developed a very powerful and very interesting biological system, which is horned beetles. He was able to make a postdoc at the University of Arizona with this, where he developed a lot of the tools he would need to develop this horned beetle system. And he later on as a professor at Indiana University at Bloomington, and he's being there. That's where I went to work with him. And he is all around great scientific and great person. It's really fun to work with him. And it's really fun to him. My first time, I knew about him by reading papers for when I was reading my own PhD defense. And then I saw him at the conferences, Evo Devo, and I ended up working with him after seeing an ad on an email list. So, all of that can happen. So without further ado, I let you with Armin Mochik.
Thank you very much for the kind introductions. Can everybody see and hear me okay? Does that work? Very good. Okay. So thanks for having me. These are complicated times, but I think one of the positives is that we discover how relatively easy it is to get together despite geographic differences. And I'm thrilled to be able to give a presentation to this class and to whoever else is listening in. And it gets recorded, and we can share it even beyond the moment, which is just fantastic. And I hope that once all of this is over, and it will be at some point soon, we will actually continue to take advantage of this. I really welcome this change to how we run and disseminate science in times of COVID. What I would like to do today is share with you a presentation that is meant to introduce you to evolutionary developmental biology, starting with sort of a very personal, let me get my few things here ready. There you go. What I want to start out with is explaining to you how I got interested in this discipline, because some of these routes were somewhat circuitous. And I developed these interests at an age that I think is not too different from the age of many of the students who are currently in this class. And then in the second part, I would like to convey to you how I think Evo Devo has begun or started out changing the way we think about the origins of organismal diversity by introducing you to what I think are the two most important technological breakthroughs that were necessary to launch us along this journey. I'll highlight one key discovery, which is Hox genes and their functions. And then I'll explore how this has begun to change how we view homology, sameness, descent with modification in organismal evolution. And then I'm going to switch gears a little bit and focus more concretely on how understanding how development works impacts your understanding of how evolution works and how developmental mechanisms bias, facilitate, limit, or otherwise influence the way evolution unfolds. And I want to introduce you to what I call the 4 H's. So this here. And I'll end with a very personal account of where I personally see the current frontiers of Evo Devo, where we're currently stuck and where we need to get unstuck in order to make progress. Now, in all of this, I welcome questions at any point. There'll be breaks in this presentation where I will ask for your questions, but you're totally welcome to interrupt me at any point. I can't follow the chat, but Ed will, and Ed knows to interrupt me and let me know if there's something that doesn't make sense or an important question came up, or if there are some technological issues. So Ed, just let me know whenever that's necessary. All right. So how did I get interested in this discipline? When I was a master's student and an early PhD student, one of the overarching questions that intrigued me concerned the origin of novel complex traits in evolution. The first eye, the first time organisms were able to see light. The first wing, the first time organisms were able to take to the sky. First time life crawled onto land out of water. The first placenta, you know, all these major innovations in evolution that launched life into new dimensions. I was intrigued how these things start out. How do you go from the before to the after? So when I was an undergraduate student, I was excited because I was able to take evolution classes and learn about potential ways to address these questions. And that introduced me to a diversity of alternative frameworks for understanding the evolutionary process. So for instance, I got introduced to a framework that prioritizes natural selection, that views organisms basically kind of like Swiss Army knives, where each feature, each trait, each part is honed by long periods of selection, shaping it to be optimized or as near optimal as circumstances allow. But I was also introduced to frameworks that prioritize history, accidents, drift, that emphasized that yes, adaptation and natural selection matters, but what really determines why or why not evolution follows a certain path are things like major catastrophes and accidents that wipe out certain parts of the evolutionary tree and allow others to flourish, regardless of how well adapted one or the other has been. And then I got introduced to perspectives that emphasize developmental constraints. Yes, natural selection matters, accident matters, but what really shapes the history of life on this planet is what development allows, the constraints imposed by engineering. Pigs will never fly, and therefore this is just fundamentally what shapes the history of life and how it unfolds on this planet. Okay, now I want to emphasize that these frameworks are all really important. They are highly influential to this day. They're non-exclusive, so they all can be accurate or relevant at the same time, but they're all equally useless when it comes to understanding innovation and the origin of novel complex traits. Why is that? It's because selection cannot select for something that isn't already there in some shape or form. It can only select for traits that have some precursor in existence. Accidents can only sort among pre-existing variation, and developmental constraints can only limit, but they do not create options. So in combination, this left me somewhat frustrated because it taught me that the origins of the first eye, wing, light-producing organ, the colorful patterns that scales make on the two-dimensional canvases that are butterfly wings, the turtle shell, the elephant's trunk, we really don't have a good framework for approaching the origin of these structures. In fact, conventional evolution biology is so stuck that it has stopped asking that question altogether. But at the same time, I got introduced to evolutionary developmental biology, or Evo Devo for short. And there's debate among many as to exactly when and where Evo Devo got started, but I'm going to put one of its major starting points in the 1980s, when people who used to not talk to each other started collaborating, developmental biologists, systematists, paleontologists, and with a shared focus on exploring how understanding development, how organisms transform themselves during their lifetime from egg to adult, how understanding that process might help you understand why and how evolution across generations unfolds the way it does, and with a specific emphasis on the origins of novelty, the origins of life, and major transitions in evolution. So once I heard about this, and 1980s wasn't too much ahead of the early 1990s when I got started as a master's student. So it piqued my interest. I want to emphasize right here that where I am now, Indiana University, is actually one of the birthplaces of this discipline. Rudy Raff, Beth Raff, and Tom Kaufman were all major contributors to, foundational contributors to this discipline, and they all worked here. All right. I think now looking back, everyone would agree that Evo Devo is a transformative addition to evolutionary biology. Many historians and philosophers of science go actually a step further and say that evolutionary developmental biology is engulfing traditional evolution in this sort of the descendant discipline that is leading in our investigation of the phenomena we're most excited about. Okay, so that's a brief summary of my personal journey to that field. What I want to focus on next is how Evo Devo changed the way we think about the origins of diversity. Where were we in our understanding and our perspectives prior to Evo Devo? Where are we now? And what were the key events that got us to where we are now? And I want to highlight two technological breakthroughs that were absolutely critical, in my opinion, at least one key discovery, and then the collective impact of both of these developments. So let me set the stage by sketching out how I view the mindset prior to the 1980s. This mindset viewed developmental biology as a discipline that describes the proximate mechanisms by which organismal form is generated from an immature egg to the final adult, how during one's lifetime an organism transforms itself. Whereas evolutionary biology was considered the discipline that describes the ultimate mechanisms, selection, drift, migration, what have you, by which organismal form changes from one generation to the next. Now, these evolutionary changes in form, they can be described in terms that are independent of how we describe the development of form. They're operating on different levels of organization, if you will. And the major implications of this mindset was that both disciplines don't really help each other out. They don't really inform each other. They don't really depend on each other. One can study the evolution of animal form perfectly well without knowing anything about the development of animal form. Plus, developmental processes are likely as diverse as the phenotypic diversity they create. So what do I mean by this? Look at these 5 photographs here. They all depict various kinds of outgrowths. Like on the left, we have the siphon of a tunicate. In the center here, we have the tube feet of echinoderm, antennae and legs of a cockroach, or the horns of a beetle, tail of a mouse. Yes, they are projections, but I mean, these come from different phyla. What could they possibly have in common? Phenotypic diversity is so enormous, so overwhelming. So must be the diversity of developmental processes that creates that diversity. I want to emphasize here that that mindset, that perspective, was entirely intuition-based. There weren't really any data available at the time that would allow us to make a statement for or against it, but it made a lot of sense, right? Okay. So now we enter the 1980s, and a couple of key developments unravel. 2 major breakthroughs happen. The first one being the invention of Sanger sequencing. Wow, for the first time, we can determine the nucleotide sequence of DNA. It's cumbersome, it's a lot of work, but we can do it for the first time, developed by Nobel laureate Frederick Sanger, 1977. PCR followed a few years later, allowing the amplification of DNA sequences to such quantities that we can now, say, visualize them along a gel, for instance, as long as the flanking regions are known well enough. This was invented by biochemist Kary Mullis, 1985. And then we had one major scientific breakthrough, I think, that was absolutely key in launching us to where we are today. And that was the discovery of the anterior-posterior patterning mechanisms of the Drosophila embryo and the specification of segment identity. If this is something you don't think about every day, let me briefly walk you through it and set the stage so to make sure we're all on the same page. This was work done by or led by Christiane Nüsslein-Vollhardt and Eric Wieschaus in the 1980s. Many people contributed to this, especially in the subsequent years, resulted in the Nobel Prize in 1995. So here I'm showing you a cartoon version of a Drosophila adult, where up here you can see the head, 3 thoracic segments, T1, T2, T3, and 8 abdominal segments numbered A1 through A8. And it's oriented from a left to right, front to back, anterior, posterior orientation. And now I'm showing you the exact same animal at an earlier developmental stage, the embryo. And we see a parallel organization. We see what will become the head, and then we see T1, T2, T3, the 3 thoracic segments, and then A1 through A8, the abdominal segments. And superimposed on these segments are the expression domains of a series of Hox genes: labial, proboscipedia, deformed, sex chromosomes reduced, antennapedia, UBX, ultrabithorax, abdominal A and B. And they are expressed in partly and sometimes not overlapping domains along this same anterior-posterior axis. And what came as a complete surprise is that that same order of expression that is visible along the AP axis is largely retained along the chromosome in which these genes are integrated, suggesting that they may have evolved by gene duplication followed by subsequent subfunctionalization. Why do we call them Hox genes? Sometimes Hom genes. This is for two reasons. Number one, when they lose their function, it results in homeotic transformations. You can think of this as one segment taking on the identity of another. And also because they contain a particular short sequence called the homeobox, a highly conserved sequence within part of the coding region that is responsible for allowing Hox proteins to bind to DNA and act as a transcription factor. So what are homeotic transformations? Here's one of the most famous. Flies are in the order Diptera because they have one pair of membranous wings. The second pair of wings is reduced to these hull tears or balancing organs. They're located in T3. And if you look at T3 and the embryo, you see that it's characterized by the expression of antennapedia, but also UBX, ultrabifhorax. And it is ultrabifhorax that is responsible, as we now know, for turning the hull tear into what it is. If you compromise ultrabifhorax function, antennapedia takes over and turns T3 into just a replicate of T2, and you get a beautifully four-winged fly. This is perhaps one of the most spectacular or elegant homeotic transformations, but it turns out that homeotic transformations were known for a long time. In fact, the term was, I think the term, I'm not 100% sure, but I think the term was coined by William Bateson. It's this slightly constipated English gentleman over here who gave us the term genetics and who cataloged homeotic transformations across phyla. Here, for instance, is a sawfly that he found that had an antenna transformed to a more leg-like structure. And then here are frog vertebrae, where he found, for instance, an individual where the normal wild type neck vertebra here that's not supposed to have any projections got transformed into one that does. Or over here is a tail vertebrate that should have projections, got transformed into one that doesn't. Okay. So the phenomenon as such was known, but the work of Nusslein Volhard and Eric Wieschaus and others catapulted us into a more mechanistic understanding of the underlying developmental mechanisms. And of course, it immediately resulted in that next question. These Hox genes, that anterior-posterior patterning of the axis, their conservation along the chromosome, is that just a fly thing? Or is that like an animal thing? Is that something we also find in organisms? And you can imagine those were thousands of studies and person hours invested into this, but long story short, the answer is yes. So I'm showing you here a mouse embryo, and this could be you when you were an embryo, where you find a similar conservation of the anterior-posterior expression of corresponding conserved Hox genes. And in fact, we find a similar conservation across various groups of organisms. Wherever people looked, it's not identical, but it's surprisingly parallel. As if animals as diverse as urchins, flies, humans are built by the same developmental genetic toolkit. And that was really a revolution, because up to that point, we thought animals as diverse as urchins, flies, and humans, surely they ain't got nothing in common. And this work suggested, no, they may actually have a lot in common. Now, I want to foreshadow, does this mean that all these body regions are homologous across phyla? These body regions instructed by the same or characterized by the expression of the same Hox genes are homologs. For now, I don't want you to think that way. For now, I would like you to consider that what this means is that homologous genetic tools are used to carry out very similar functions, in this case, organize the body along the AP axis in very different organisms. Okay. It didn't stop at Hox genes. It got crazier and crazier. Example number one, eyes. We know that the eyes of insects, squids, and humans represent examples of independent convergent evolution. They're not homologs, but homologous transcription factors, opsin proteins, cell types, and neural circuits are used in all of them. Hearts. The hearts of insects and the hearts of vertebrates are independently evolved contractile pumps. They're not homologous structures, but homologous transcription factors and cell types facilitate their formation during ontogeny. Remember these 5 images at the beginning? 5 projections, outgrowths, sticky outies, as we sometimes call them, across phyla. They're definitely not homologous structures, but they utilize homologous transcription factors to specify the proximal distal axis. The same homologous transcription factors are used to determine where each of these structures embeds itself into the body wall, where it outgrows, and where the medial regions are for each of them. So what are the implications of this? Well, it suggests while organisms are indeed incredibly diverse phenotypically, how they are made during development may actually be rather conserved. The same genes, pathways, morphogenetic processes are used over and over and over again to make both similar and different traits in very different organisms. Organisms and their parts emerge more like Lego creations. A lot of diversity and novelty emerges in this process, emerges by reusing and recombining the same parts in different ways. You make new things not by creating new Lego blocks. You make new things by taking old Lego blocks and putting them together in different ways. How's that? Took me a while to find the Lego mantis, but here it is. Okay. So in a nutshell, clearly non-homologous traits are built using homologous genes and developmental processes. Okay, now I'm going to mess with your brain because I'm going to try to convince you that the opposite is also true. Homologous traits can be underlain by non-homologous developmental processes. Okay, what do I mean by this? One of the best examples concerns insect wings. Insect wings originated only once. There's only one common ancestry of insect wings. A wing is a wing is a wing across insect orders. Doesn't matter. But different orders differ quite remarkably in how they make their wings during development, from timing to morphogenetic movements to developmental pathways that are employed and how they're interacting with each other. Okay, taken together, Evo Devo therefore forced a transformation of our understanding of sameness, homology, what it means to have 2 traits in descendant lineages. It used to be black and white. You either were or you weren't homologous, end of story. Thanks to Evo Devo, it's now more complex. Homology comes in layers. It can be partial, it can be deep. It opened up the possibility to learn from development why evolution unfolds the way it does and not some other way. And it allowed us to assess our own assembly. All of a sudden, we could look at ourselves as a Lego creation and our own making in development and evolution. If you're interested in how the human heart forms and why it sometimes malfunctions, studying the insect heart all of a sudden is a possibility. It wasn't before. But we also had to grow cautious about accepted model systems. Just because something is phylogenetically close, like the mouse or the rat, it doesn't necessarily mean that its development is similarly close to our own. Thalidomide affects appendage formation in humans in extraordinarily debilitating ways. A mouse doesn't give a damn. You can hit it with orders of magnitude higher concentrations. The way appendages are initiated during mouse embryogenesis is just far more robust to that perturbation. Collectively, it brought us significantly closer to understanding how development facilitates the evolution of organismal diversity and novelty. And this is what I'm gonna focus on in the next part of my presentation, but I want to stop here for a moment and see if there are any questions or comments, or if there's anything that didn't make sense that I touched on or you'd like me to elaborate upon. And it allows me to get a sip of water.
We have one question in the chat. I don't know if you wanted to say it yourself, since you're connected.
I actually can't see the chat right now because I'm sharing my screen and my chat is, I'm not sure where it is.
Okay. I was just curious how we can talk about structural convergence if the genetic mechanisms behind is homologous. Do we need two concepts of homology depending on the level of analysis?
So if I understood this correctly, the question was, how can we establish homology between traits if the underlying developmental mechanisms aren't the same? And that is a great question. And I'm actually at the point where I think that the term homology, just like the term novelty, has sort of lost its relevance. To me, it's all descent with modification to varying degrees and at varying levels of biological organization. I think we can establish homology if we are absolutely precise about the level of biological analysis that we're talking about. Is it genes? Is it gene networks? Is it hormonal mechanisms? Is it cell types? Is it complex morphologies like organ systems? And then I think it's an informative discussion. But the notion that an entire trait either is or isn't homologous, we're done with that. That's biologically just no longer realistic.
Great, thank you.
All right, should I go on?
Yes, please.
All right, so in the next part, what I would like to focus on is what have we learned since? What are some of the key developmental mechanisms that affect evolutionary change in a way that is of interest to an evolutionary biologist, because it helps us understand why and why not evolution took a certain route over other possibilities. And I want to introduce you to what I call the 4 H's. What are the 4 H's? The 4 H's are heterotopy, heterochrony, heterometry, and heterocyberny. What do we mean by this? Okay, let me walk you through it in the abstract, and then I'll give you examples for most of them. So heterotopy is an evolutionary change in developmental location. Heterochrony is an evolutionary change in the timing of development. And heterometry is an evolutionary change in the amount of a developmental product that you produce. Heterocyberny is the recent addition to the 4 H's, and it's an evolutionary change in the governance of a particular developmental process. And it turns out that I am one of, I think, 2 people on this planet who uses the term heterocyberny. So I'm going to leave it at that and focus on the other 3. But the phenomenon itself, evolutionary changes in the governance of a process, bringing something old under the control of something else that it hasn't been under the control of. I think it's a somewhat understudied Evo Devo mechanism that deserves further attention. But what I want to focus on is heterotopy, chrony, and heterometry. All right. Examples of heterotopy. It simply refers to an evolutionary relocation of an old developmental process into a new developmental context, location. For instance, the developmental machinery that establishes the proximal distal axis of leg formation or appendage formation is A, well understood, and B, phylogenetically ancient. That doesn't stop it from being reused in the evolution of novel outgrowths, like beetle horns, a much more recent invention during insect evolution. Not all of them have been recruited, not all to the same degree, but if you wanted to evolve beetle horns and you needed a machinery that sets up a proximal distal axis, you didn't need to evolve it de novo. It was already there. All you needed to do is find a way to get it into this new location, anthropomorphically speaking. There are hundreds of examples like this right now in the Evo Devo literature. Heterotopy is a super well-established mechanism by which relatively simple changes in development can have profound evolutionary outcomes. Now, it's also not restricted to adding old parts to new places. You can also add old functions to new places. So you can take your beetle horn And now you can recruit this pathway, the double sex pathway. This is an ancient pathway that determines somatic sex, whether, for instance, ovaries or testes develop in a female or a male. Recruiting it in the context of beetle horn formation, you can now create horned males, but hornless females. Or recruiting the hedgehog signaling pathway now allows you to make that same development nutrition sensitive, allowing you to form big horns in large males that had a lot of food as developing larvae, but hornlessness in small males that instead engage in alternative sneaking behaviors. So again, profound evolutionary outcomes enabled by relatively simple changes and simple relocations of old, ancient, if you will, developmental pathways. Here, perhaps the Lego analogy is most appropriate. You take old blocks to build something new rather than evolving new blocks each time you create a novelty. What about heterochrony, evolutionary changes in developmental timing? That's perhaps even easier to envision. Heterochrony-mediated diversification in relative appendage size is found all over the place. The reason bats have relatively long forelimbs and dinky hind limbs, or the other way around, and lots of flightless birds, it's because the timing of development of these enlarged appendages simply starts earlier or continues for longer than their counterpart at the other end of the body. It's a simple change in when you turn things on and off that can have profound changes in final phenotypic outcomes. They can also be even revolutionary. Here I want to introduce you to heterochrony-mediated origin of complete metamorphosis. Now, let me tell you up front that right now this is more a hypothesis that is receiving growing support than fully well established, but I think it's getting there. So here's what you need to know. Entomologists divide insects into two major groups, if you will. Insects that have what we call hemimetabolism, or incomplete metamorphosis. Think of a grasshopper or a cockroach, where an egg hatches into a nymph that looks kind of like the adult, just much, much smaller. That nymph molts into progressively larger nymph that then turns into an adult and is done. And that's in contrast to holometabolous development or complete metamorphosis. Think of a beetle or a bee or a butterfly, where the egg hatches into a larva, which looks nothing like the adult it will eventually turn into. Think of a caterpillar and a butterfly. They look nothing like each other. That larva, again, molts through a couple of instars into progressively larger versions of itself, then molts into a pupa, and then molts into an adult. And so a lot of debate surrounded and we know that hemimetabolous metamorphosis is the ancestral state. And so a lot of debate surrounded the question as to how you go from hemimetabolous to holometabolous in development, in evolution. In particular, how do you evolve an entirely new life stage like the pupa? Because the understanding was always that, well, an egg is an egg is an egg. Eggs gotta be homologous to each other. Adults gotta be homologous to each other. And well, it kind of looks like that nymphs got to be homologous to larvae, and that leaves the pupa as this brand new stage that was somehow invented by holometabolous insects. Seems like a tough thing to do, though. Now, what I didn't tell you is that once people looked closer at the very early stages of hemimetabolous development, they found that there's another stage called the pro-nymph. The pro-nymph is a very short stage. It can last often just hours, and often it is spent entirely within the egg. So you actually don't see it externally, but it is a separate molt. And if you look at a pro-nymph, it looks nothing like the adult it's going to turn into. And now a growing body of evidence provides support for the alternate hypothesis that yes, eggs are definitely homologous to eggs, and adults are definitely homologous to adults. But what happened in the origin of holometabolous metamorphosis is that the pro-nymph has been elaborated to give us what we now recognize as the larva, and the nymph has been contracted to what we now recognize as the pupa. So here we have a heterochrony mediated change that transformed, revolutionized insect evolution by decoupling larval from adult development in ways that allowed insects to radiate into niches that were previously inaccessible to them. All right. Lastly, heterometry, evolutionary changes in the amount of developmental product. Again, this is actually developmentally relatively easy to envision. You just make more or less of something. For instance, you make more of BMP4 or calmodulin or less, and therefore your beak is going to be longer and curved or shorter and stouter. It is the interplay between these two transcription factors that affect the morphogenesis of beak development. If you want to evolve a novel beak, or if selection favors the evolution of a novel beak shape, selection does not need to come up with new mechanisms to specify these shapes. They're already built into the making of the very first beak. All you have to tweak with is the relative quantity of a certain developmental product. Cool. Now, that raises the next question. So which mechanisms then enable developmental processes like amount, location, timing to be so modular in time, space, and production? How can these things be changed so easily? Well, what I want to convince you is that the processes that regulate how much of something you make, or where you make it, or when you make it, these processes are themselves modular. They act in a combinatorial manner, and they facilitate the evolution of diverse and novel regulatory settings without having to evolve novel regulatory machineries. Oh my God, what the heck am I talking about? Let me break this down a little bit, maybe with the help of an example. All right, let me see if I can make this work. So here, for instance, is a cartoon version of the hedgehog signaling pathway. The hedgehog signaling pathway involves a diffusible morphogen called hedgehog that binds to a receptor called patched that is normally inhibited by another membrane boundary molecule called smoothant or protein. And when they're bound or not, they affect a single transduction cascade in the cytoplasm that allows this protein here, cubitus interruptus, to enter the nucleus, either cleaved or not. And then it turns certain genes on or it doesn't. And that pathway is itself incredibly conserved. Doesn't matter whether you're looking at a fruit fly or a sea urchin or a mouse, it's the same darn pathway. Nothing ever changes. What changes, however, is what hedgehog excretion is responsive to. What changes is the kinds of genes that this pathway turns on or off. The inputs and outputs are easily changed, dissociated from each other via a highly conserved signal transduction machinery. The same is true for transcriptional regulation, for instance. Here, the basic machinery, the transcription factors and cofactors it takes to initiate transcription is highly conserved. But the combinatorial interaction between them enables a relatively small number of elements to regulate thousands of distinct decisions. So what I was trying to explain is the combinatorial reuse of modular conserved elements across contexts facilitates the evolution of diverse and novel regulatory settings without having to evolve novel regulatory machineries. Okay, let me step back and try to summarize this a little bit. So our conventional perspective at the onset has been that knowledge of genetic variation and how it changes and is changed by selection, recombination, drift, migration, is sufficient to understand, reconstruct, and predict phenotypic evolution. This is what I learned in my introductory evolution classes. What did Evo Devo contribute? Evo Devo contributes that an understanding of the modularity and dissociability of developmental processes creates degrees of freedom along which developmental evolution can proceed more easily than others. It creates landscapes along which evolutionary processes are allowed to operate. Diversity is facilitated not despite this conservation of development, it's facilitated because of it. Lego creations are incredibly robust because their fits are incredibly conserved, not despite the conservation of individual building blocks. Now, this Evo Devo perspective doesn't mean that what I learned in my basic evolution classes is wrong. Of course, all these processes matter. All that Evo Devo says is that it's not all that matters. If we want to understand why and why not evolution unfolds the way it does, understanding how phenotypes are generated in development provides valuable insights. Okay, I want to stop here for a moment and see if there are any questions or comments or thoughts.
We have none in the chat. I don't know if anyone wants to ask something. Just move on.
I have one. You show at the beginning of the presentation that there is a correlation between the gene order, the Hox gene order, and the expression pattern in the individual, in the anteroposterior order of the individual. To my knowledge, there is no genetic basis to understand why that happened, why there is a synteny so preserved in all the different phyla. Do you know why that is a fact?
So I agree that this came totally out of the blue. This was not predicted nor expected, but it creates the possibility that the Hox code in its initiation originated by a gene duplication, and that as Hox genes were added through gene duplication, so was a genome's ability to specify distinct body regions, either by bringing them under the purview of particular Hox genes or Hox gene combinations. I think Ed might be better equipped to address that question.
So why the complete mechanism, why it's that way, is not really known. It is known that you can have functioning Hox clusters without conservation of order. Also, something important, normally we think the spatial pattern of expression, but normally Hox also temporally expressed in that same order. So there are a number of hypotheses about why that is the case. Some say that it has to do with the regulatory sequences that are interspersed between the genes. And so that the fact that you basically are reading along the chromosome will turn on or off inhibitors from the previous Hox genes. And so any kind of explanation has to then deal with what happens when of synteny conservation is not the case. But the problem is that, you know, you can break free of that constraint if you find a different way of regulating. And so what might happen is that you have a basic initial way of regulating Hox genes through their order in the chromosome that many times have changed, and then that results in then once you change regulation, then you're no longer constrained to keep your Hox genes in order in the chromosome. But there's a lot more hypothesis than actual strong data, even now.
All right, thanks, Ed. So what I wanted to end with are just a few slides that are now again fairly subjective. They highlight where I see the current frontiers of this field. It's a very rapidly moving field, which is always a good sign that exciting things are about to happen. Here is where I see some of the greatest challenges. Number one, and some of these we're beginning to meet and others we have yet to really fully confront. Here's one. So extending Evo Devo research to non-model systems and structures. That is well on its way. It used to be that if you wanted to do Evo Devo, you were limited to the fly, the worm, the mouse. We're done with this. You now routinely see Evo Devo research done in such unusual organisms as butterflies and their wings, horned beetles and their horns, stag beetles. Here is some of our own work on fireflies. And this, by the way, is actually a Hox gene knockdown, which not just deletes the light-producing structure, this lantern, but it also converts genitalia into legs, which was really an interesting, I mean, it was to be expected, but I didn't think it would work so well. All of this is helped by the growing availability of key methods like RNA interference, CRISPR-Cas9, but also the growing ease of large volume sequencing. You can do next generation sequencing on any organism you want. And you can do RNAi on, well, nearly any insect you want. And Evo Devo now favors the kind of researcher who knows his ecology and his behavior and his evolutionary history really well, because now you can select perhaps the best organism there is to address whatever behavioral, physiological, ecological question you have in mind. You're no longer limited to just a handful of established systems. Here are some vertebrate examples of recent models in Evo Devo, from marsupials to chameleons to the jerboa down here, the crazy, just a crazy Dr. Seuss kind of creature. Okay. A second frontier is the integration of Evo Devo with ecology. I find this perhaps the most critical integration that needs to happen, because we know that how development unfolds is impacted by ecological conditions. It's impacted by environmental conditions, and it's impacted by environmental conditions that organisms themselves sometimes shape. And it is this interaction that determines the kind of phenotypic variation that evolutionary processes then see or don't see and can act upon. And this has now given birth to what is sort of the newest descendant discipline, ecological and evolutionary developmental biology, or Evo Devo. To me, that is one of the most exciting areas, I think. And if I were a beginning grad student now, this is one of the areas I would consider throwing myself at. I think we need to think hard about what we consider the environment, reevaluate what we mean by it, and reassess its potential role in evolution. Traditionally, we view the environment as something that is external to the organism. It's separable and it's passive. You take the organism away, the environment is still there. That's how I grew up. But we've now learned that environments, internal and external, are shaped by organisms themselves, often to significant degrees. Where the organism ends and where the environment begins is often not as easy to tell as we might think. In fact, aspects of the environment are often inherited in ways that may affect evolutionary outcomes, whether it is the transmission of microbial symbionts from mothers to offspring in the context of dung beetle, or the curation of entire ecosystem by dam-building beavers, or, well, the creation of environments that surround us, that have fed back on our own evolution in profound ways. Reassessing the role of the environment in development and evolution, I think, is overdue. So what I'm going to end with is this. Evo Devo continues to be one of the fastest transforming disciplines in biology. What we thought to be true 15 years ago has often already gone out of the window. In fact, what we thought to be true 4 years, I'm scooping myself left and right, trying to publish things that I am beginning to realize with the newest data are probably not correct. This is a great sign for, if you are an undergraduate or a graduate student thinking about which area to go, you want to go into those areas that continuously shoot themselves in the foot because they have to revise findings that are just a few years old. And so with this, I want to thank you for your attention and welcome any questions and any comments and any suggestions anyone might have. So thank you.
Oh, it's great. So do we have anyone in the audience? I have a couple of questions here on the YouTube chat. So Tomas Gonzalez is asking, there is some sort of functional correlation between heterochrony changes and heterometric changes?
Oh yeah, you know, that's a great question, and I should have emphasized this. These 4 H's, they don't need to act in isolation. They can act combinatorically. And just imagine you simultaneously change location, timing, and product, and then bring all of this under the control of something else. Each one of those is a relatively simple developmental change, but the phenotypic output is dramatically different from where you started. So yes, they can absolutely act combinatorically. Off the top of my head, because my brain's a little tired right now, this is my second lecture for today, I can't think of one, but I'm sure they do exist. I want to add one more thing, and that is more to the beginning of my talk, and in fact, Ed's introduction. Ed mentioned that he came to my lab by responding to a postdoc ad that I published. There's a good chance a similar ad will go out pretty soon, as we're looking for a new person, if the funding situation works out well, but I'm quite optimistic. So if there's anyone out there who is interested in this kind of work, talk to Ed about his experiences, learn more about what it's like to maybe work here, and then I'll make sure the ad and any other opportunities that we can offer get disseminated in your directions. I don't know if this is obvious to you already. It's sometimes hard to see if you say an undergraduate student, but Evo Devo is fundamentally an interdisciplinary science. Science is a fundamentally international affair. We are a lab of immigrants. Americans are in the minority in our group. We have currently, I think, like 6 or 7 different languages established in the lab. I have learned that it takes diversity of backgrounds, biographies, etc., to get a really good overview of a particular phenomenon. So don't let going to a foreign country for graduate work or postdoctoral work ever hold you back. I think it's an incredibly rewarding and eye-opening experience, not just for you, but also the people you end up working with.
Yeah, I can totally attest to that. I'll be happy to talk, counsel, mentor anyone who's interested now or in the later future to study abroad. There's a lot of things to be done. There's a lot of interesting things to do. People who will be happy to receive you and teach you and have you teaching them. This is always a two-way lane. It's you give. It doesn't matter if you're the student or the mentor or the professor. You're always giving and receiving a lot. So don't be afraid to move around.
A couple more things. So I have never taken a developmental biology class in my life. Yet here I am studying development. What I didn't talk to you about today is we have a research program on host microbiome interactions. It's in the context of Evo Devo and how interactions between hosts and symbionts and the fact that organisms often form teams allows them to innovate in ways that individual taxa cannot. I've never taken a microbiology class in my life. I don't know what I'm talking about. That doesn't stop me from doing this kind of research. And a lot of these directions were initiated by people coming to the lab with an expertise I was lacking, or educating me in ways that I needed to then think about opportunities that my organisms offered that I wasn't aware of. Now, I saw in the comments one comment that said Evo Devo sounds amazing. I agree. And I think now is the time, because more than ever, perhaps, it is a little bit depressing, but it's also an enormous opportunity. Nowadays, we never had such a number of species that are facing a profound changes in their developmental environments. If anyone is interested in studying the interplay between ecology, environment, and ontogeny, development in evolution, now's the time. And because if you look at global climate change, if you look at invasive species, if you look at species migrating as fast as they can, now is the opportunity to explore how that interplay determines where evolution can and cannot go.
Great. We have one question popping up from the YouTube channel, but I think I hear, so if you want to ask it.
Thank you. Well, the question is, do you think the evolutionary impact of the developmental mechanisms can be understood ultimately at the level of change in gene frequencies?
Can it no, because the so that's a great question. If I'm very optimistic, then I might say maybe eventually at some point, and I would say that because it would require that our understanding of what genes do and how they influence phenotypes is understood. But for the most part, we really don't. And for the most part, the phenotypes we care about are the emergent property of processes that are contributed to by so many levels of biological organization, genes only being one of them. And so there are some clever analogies that you could ask by, would you ever understand the meaning of a metaphor in a poem simply by understanding all the letters that your poem is made up of? No, of course not. It takes levels of organization in between that are needed to convey that phenotype, if you will. But what I would like to emphasize is, and maybe I should have listed this as another frontier, a lot of Evo Devo is genetic in nature. And to the extent to which it's genetic in nature, it must obey the rules and limits and laws that guide population genetic processes. There's no way around it. And what we are still lacking is a marriage of population genetics and Evo Devo. We're beginning to slowly get there, but how developmental evolution unfolds on the level of populations, we actually don't know so well. Relatively few people have begun to study that. And there's a lot more work that must be done before these two perspectives can be married.
Right.
Related with that answer, last Friday we have a philosopher and we discussed concepts of inherence, and he concluded that there is no vertical inherence, then it makes no sense to speak about natural selection. And you talk about niche transformation, but that can occur horizontally. So what do you think about the concept development or that the Evo Devo brings to us like niche transformation in the context of the evolution and the natural selection?
So I'm not sure I fully understand the question, but let me, would you mind rephrasing it real quick?
He concluded that there is necessary a vertical inherence to make sense of the natural selection, and he said that any horizontal inherence or any horizontal transformation like niche construction is outside of the natural selection.
Okay. I get it. Okay. Well, first of all, I think you can have natural selection no matter what, but evolution by natural selection requires vertical inheritance of some way, shape, or form. And especially if you define evolution as a change in gene frequencies within a population. Now, for the moment, consider expanding that definition a little bit and consider evolution and a change in the heritable variation in a population, genes and beyond. Okay. Now, a lot of environments that organisms construct exhibit heritable variation, or at least are heritable, from mRNAs to hormones that mothers pass on to their offspring, to vertically transmitted symbions, to positions in a social hierarchy or territories. Now, that inheritance, I agree, it happens over probably a short number of generations, but a growing number of models show that it doesn't need to last for a long time to have an evolutionary impact. The same goes with learned knowledge. Any form of cultural transmission, even though it may just operate over the tens or hundreds of generations, can lead to profound divergence between lineages. So I agree that you need to have vertical transmission of whatever, and you need to have variation in what is being transmitted to have evolution by natural selection. I would agree with that, but I don't agree that this somehow limits you to genetic inheritance. That said, this is part of a much larger topic area that Ed knows a lot about. I'm one of a whole bunch of people who have been active in the context of putting the extended evolutionary synthesis to the test. If this is a topic area that is of interest to this group or whoever, I'm happy to talk about that separately. But my brain needs a little time to prepare for that, because I need to rethink my head for some of this so I don't say something stupid.
All right, we'll take that and figure out another time to speak about that. I have one question in the chat from Lican Martinez, asking, what level of impact do you think development has in shaping evolutionary trajectories? And he says he knows its accepted role is now growing, but how do you think, how protagonist will it end up being in the next few years of evolutionary theory?
All right, I see that question too in the chat now, and it focuses on the role of developmental bias in evolution and our understanding of that concept. So let me answer it the following way. I think that whether or not there is developmental bias in evolution is no longer a relevant question. We're done with that. Evolution can only do or will do what development shapes, biases, enables it to do. The relevant question is exactly how does development structure phenotypic variation that is available for evolutionary processes to act upon? Under what circumstances, in what contexts is that structuring limiting or facilitating? And it's sort of in the eye of the beholder. If you call something a bias or a facilitation, there is a big gray area. And the last thing I want to add to this is, if you give me a null model, if you tell me what should be happening in the absence of developmental bias, then I can tell you whether there is or isn't bias and whether it's facilitating or hindering or limiting. But it's hard to envision any kind of evolution, any kind of phenotypic change occur outside the context of, outside some kind of developmental setting. By virtue of being, for lack of a better word, by virtue of being intrinsic in development, development must somehow bias, structure, delineate phenotypic variation. Here's a shorthand that often works for me. Mutations may be random within a genome. What organisms do with them is not. What developmental systems do with the mutational variation they receive is not. It is channeled in some way, shape, or form by developmental systems into phenotypic variation that either is visible to selection or cryptic. Now, whether that channeling is biasing or not, that's an empirical question that needs to be asked one trait and one level of biological organization at a time.
I have a question. Good afternoon, Armin. Thank you for your presentation. So about the 4H you mentioned is about the heterotropy. It's very difficult to me to understand one example or to visualize one example about the process to the all functions.
Say again, please.
Yes, the process to add all functions to newly position.
Mm-hmm.
It's very difficult to me to visualize an example. If you can give me one, please.
Okay, so the example I try to give, let me try to re-give it, is the double sex signaling pathway, which is a single transcription factor that, however, has alternative splice forms. Its ancestral function is to promote male development of genitalia in males and female development of genitalia in females. The moment you invent that mechanism, the moment you can, within the same, almost the same genome, certainly in the same species, have a switch that says, make a male-like structure in this set of circumstances and a female-like structure in this set of circumstances, you are no longer restricted to just doing this to genitalia. You can do this to beetle horns. You could do this to the context of behavior. You create you take an old function, make something sex-specific, and apply it to a new location, from genitalia to the head or wherever you want it. Am I helping? Okay. Any other thoughts or comments that people want to make?
I see no more questions in the chat.
Well, I have another. In a previous class, we discussed the Cambrian explosion and the relationship with environmental changes and the origin of the Hox genes. How important do you think the origin of these genes has been for the great disparity and diversity observed in that short evolutionary time?
Not sure I fully understand the question, but let me give it a try. So there's a rich literature in the origins of novel complex traits. And when you read that literature, you find that what people call novelty and complexity is really all over the place. One of the earliest proposed mechanisms, in fact, with respect to the origin of phyla, was by Stuart Newman and others, Gerd Müller, that suggested that pre-Cambrian, the association between genotype and phenotype was so loose that it allowed for a lot more phenotypic exploration than what is present today or possible today, where the association between phenotype and genotype is more rigid. I never really bought that argument. It made no sense to me, but it also was sort of irrelevant because the innovations that I'm interested in, the horns of beetles, the light-producing organ of fireflies, the novel appendages of scorpions, for instance, that I try to work on, the gaudy helmets of tree hoppers, all these are, by comparison, really recent. Mm-hmm. And we've had a tight association, if you will, between genotype and phenotype, and we had all our phyla in place for 500 million years. And we have all this incredible conservation of genes and developmental pathways. Think of this hedgehog signaling pathway example I brought up. I mean, we have like half a dozen of these and they are invariant. Still, we manage to innovate in evolution, don't we? We get a lot of innovation despite all this conservation. And that made me think that perhaps that's the wrong way to look at it. maybe we're not getting, maybe evolution isn't able to innovate despite all this conservation, but because of it. And this is how I try to motivate that Lego analogy, where a lot of ability to produce something new comes from the fact that what you're building it with is highly, highly, highly conserved.
Okay, thanks.
To me, it's a little bit, there is a bit of an analogy you can draw to architecture, where you can build novel forms and structures and buildings by drawing from the exact same building blocks, knowing that each time you put them together, they're gonna fit, they're gonna work together. It doesn't stop or doesn't negate the possibility for some new innovations, you know, like the first time opsons were invented and life on this planet could see and was able to perceive light. That was, you know, a quantum change that required really the invention of a new protein. The same happens in agriculture, in architecture. The moment you say, came up with steel, you could build in ways you couldn't do before. Still, that continue to allow you to take advantage of pre-existing building blocks that continue to work well with each other.
Okay, you talk a lot about animals. What happened with plants?
Who cares about plants? I don't know. There is a rich area of plant Evo Devo, and I don't want to belittle that. And there are areas where I overlap with researchers there. For instance, in our shared interest in major evolutionary innovations like flowers, for instance. Flowers are like the quintessential plant innovation, but there are many others. It is, you could think of it like phylogenetic inertia. I just didn't ever start with plants, and so it doesn't really often enter my thinking, but it should. In some areas, however, it now has. When it comes to, say, niche construction, non-genetic inheritance, plants do that left and right, where environmental experiences by mothers or grandparents affect how seedlings will develop and respond to environmental conditions during their own ontogeny. And this is where plant development actually offers a lot of experimental advantages.
Okay, we have one last question from Ben.
The last one from me. Thank you so much, Armin. Similar to the previous question, but during the talk, you mentioned that the Evo Devo, for some people, they think that is more general than the previous modern synthesis, let's say. If that statement make me ask, is the development process a restricted process, something that only happens to multicellular organisms? Or for instance, we can say that the bacterial biofilm formation is an example of the developmental process in prokaryotic organisms?
Yeah, no, it's not. At the beginning of my talk, I think I might have mentioned that, you know, one of the perennial challenges is to understand the origins of life, major transitions in evolution. And one of those, first and foremost, is what it took for cells to form collectives and to form tissues. And the study of biofilms, the study of temporary cellular aggregates falls right in there. And so in that sense, I see a continuum, but I think there's still a, and now I'm struggling to articulate that properly. I think if you want to study the trend, the evolution of three-dimensional form and function, this is something that simply doesn't exist in a bacterial biofilm in a way that would lend itself easily to parallel research programs. That doesn't mean to say that the study of single-celled organisms, for instance, their environment responsiveness, there's a lot of work done on quorum sensing genes in bacteria that I find remarkably fascinating, and that actually held a lot of insights into the evolutionary consequences of environment-dependent gene expression. There's no reason you have to do this in a multicellular organism. Absolutely.
Okay. Well, we have, I'm just gonna scan around, but there's no more questions. I just wanted to thank you, Armin, for talking to all of us. It's really cool. I actually have never had a chance to have you teaching, teaching Evo Devo in a class. So it was really cool and good experience too.
Thank you very much for having me. This works both ways. So when I teach or talk about these things, I discover let me rephrase that. When me, myself, and I talk, when I have these conversations in my head, it always makes sense to me. So talking to you or others always helps me discover where I need to think some more and where my own thinking isn't quite clarified enough to explain it to others. So thanks for giving me this opportunity. I really enjoyed it.
All right, we hope to see you again talking about the extended evolutionary synthesis.
Very happy to.
Thank you very much.
All right, thanks everyone for watching. And this is gonna be, this was streamed live on YouTube. Don't forget, this is gonna be going through the channel of the university. So don't subscribe if you haven't done it, you know when the next talk is coming. And thanks everyone for being here, for the questions, and hope to see you next time on Wednesday.
Take care, everybody.